Welcome to Making Sense of Complexity, featuring conversations with complexity science practitioners and philosophers. Our goal with this series is to share insights on how to make sense of our complex and uncertain world. On today's show, our guest is Dr. Mark Cohen, a neuroscientist and co-inventor of functional magnetic resonance imaging. Mark is fascinated by the relationships between structure and function in the human brain and intensely curious. Whether exploring the brain, the complexities of machine learning, or the beauty of musical polyrhythms and harmonics, we've got a lot to explore in this one. Let's begin.
Mark, welcome to Making Sense of Complexity. How are you today?
I'm terrific. It's a pleasure to be seeing you again, and I'm looking forward to the conversation.
Excellent. And where are you calling in from today?
I'm in lovely Pasadena, California.
The LA basin. It's beautiful there, I know. Sometimes you've had quite a bit of rain and even snow recently. How's it going today?
Yeah, there was a funny experience a couple weeks ago when I looked out my window and somebody had put a mountain there because, things look a great deal bigger when the snow is on top of them. But today we're enjoying beautiful sunshine, and I personally happen to love the water coming down. We need it.
Oh, that's great. I just want to start with a question about your background in studies of the brain, neuroscience, fMRI. What drew you into this field originally?
I had the terrific experience as an undergraduate of having a roommate who was a graduate student in philosophy. His name's Davis Baird, and he went on to be quite the philosopher. But the discussion around the house was more about Kant than about what to eat for dinner that night. And the confluence of my own already very technical disposition and learning about these deeper areas really inspired me to search for neuroscience and to try and understand meaning in life through the brain.
So you picked an easy topic, right? The brain is one of those things that is very, very difficult to look at. I mean, it's just three pounds of tissue or something like that sitting in the middle of our heads. So one of the techniques I know that people talk about a lot is functional magnetic resonance imaging, which I understand you're an expert about. So maybe you could just give us a quick description of how it works.
Well, I'm happy to say that it was an invention of my friends and myself, this fMRI, and it's a very fun tool. I'm going to give you just a 2-second about me. I studied the brain at the level of neurons in my graduate program, and this involved the slaughter of a great many animals by the time we were done with it. And I realized that the kinds of questions that we could explore with electrophysiology were they weren't addressing any of the interesting stuff. The way I put it at the time is I could put an electrode in every neuron in a rat's brain and have no idea how it feels. We just couldn't get there. So I actually left the field and decided to go into medical instrumentation. And I chose as a first target MRI. And MRI at the time was very, very, very slow. 15 minutes to get a single slice picture of the brain, and it required a lot of the people who were in there. So I set out working on the question of how we could make the tool faster. And during the course of a few years in the business world of MRI, we took the scan times down from 15 minutes to about 32 milliseconds. And at 30 milliseconds, we're now acquiring movies at kind of video rate, which you wouldn't think at first glance is all that important when you're looking at the brain. It's just a brain. It's sitting there. You look at it over and over again. And indeed, if I were to show you a movie of the brain, no matter what it is you're doing, you would say, well, there's nothing going on, right? It's just a picture of a brain. But it turns out that the signal varies. And the signal varies in the brain because of the level of oxygen that's bound to the blood that's going to any particular part of the brain. So hemoglobin is the primary carrier of oxygen, as I'm sure you and most of your listeners know. And hemoglobin has an iron atom on it. which bonds to the oxygen. And depending on whether the oxygen is present or not, the iron changes magnetic state in such a way that it creates a tiny, tiny difference that can be exploited in the MRI signal. So we watch the signal fluctuate, but it's fluctuating locally because of the amount of oxygen in the blood. Well, that turns out to depend on how much activity is going on in that particular region of the brain. So neurons require oxygen to work. The harder they're working, the more oxygen they require. And your local blood vascular system is so finely tuned that tiny, tiny areas of the brain, what we call cortical columns, are able to demand more oxygen at the moment. And we can see that in the MRI. So what we can then do is take a movie of the brain and study the fluctuations at every tiny location in that movie and compare those to what the person is doing. And what they're doing might be something straightforward. The first kinds of experiments we did were, for example, have them look at an intense flashing light. And we can see the brain demanding more oxygen and therefore, in this particular case, producing a larger MRI signal when the light is on than when it's off. But we can then go ahead and say, how about think about a blinking light? And guess what? We can see that. So the tool has become extremely subtle in the kinds of questions that we can ask. And that's what's called functional MRI.
Yeah, and is it, how finely grained is that ability to see the magnetic state of the hemoglobin cells as they're delivering or oxygen to the cells, to the neurons? How finely grained is that? Can you actually see down to the level of individual neurons?
So we can think about fine-grained in a couple of dimensions. One of them is temporally, right? I mean, the blood flow doesn't change instantaneously, but with some cool techniques and certain tasks, you can get resolution down to a few tenths of a second out of this. So it's pretty good. It's slow compared to thought, but it's pretty good. Spatially, we can get the what we call voxels, which is a three-dimensional pixel. We can get the voxel down to about a half a millimeter. And that's that resolution is close to the size of a human cortical column. And what we still don't know is if we push it even further, which of course people are trying, if we push it even further, we don't know whether the resolution of the vascular signal is any better than the size of a cortical column. So when you look at the microarchitecture of the brain, you can see local blood vessels that appear to be local to one or two cortical columns. And I don't know if you or your listeners need a little more understanding what a cortical column is to make sense of all of that.
Well, I guess the simple question is, how many neurons are there in a cortical column that particular blood flow that you're measuring magnetic resonance for, would address? I mean, how many
tens of thousands to hundreds of thousands.
Okay. Yeah. So it's still, in a certain sense, a fairly coarse graining of the actual neuronal functioning, but it gives you a pretty good idea of what areas of the brain are firing off in particular times, and in that, the fairly short time frame. What was the time frame again for that?
Well, so it depends on the use and the kind of task you're doing. Best case, tenths of seconds.
Yeah. So we have a long way to go yet in terms of diving into the guts of the brain. Are there other techniques that are coming along that are helping to get some even more finely grained pictures in fMRI?
Oh, sure. I mean, the techniques go down, of course, to single synapses, but they don't have the kind of field of view that MRI does. You can't see the whole brain at once at the level of synapses, and it's not so clear that the functional resolution of the brain for studying at least the kinds of cognitive processes that I personally am impassioned about, requires any higher resolution than we've got. To study cognition at the level of an individual neuron, I think is a very poorly framed question.
Yeah. Okay. Well, that's interesting. Let's touch on the complexity of the brain itself for a minute in terms of, the aggregates. As I said, I understand it's around 3 pounds of tissue. And can you sort of elaborate on like how many neurons are there? How many, connections does each neuron have? That kind of data.
So we don't know. When I was in graduate school, we used to talk about that there are 10 to the 10th neurons in the brain, of which 10 to the 11th are in the cerebellum. And because it's hard to count these things.
Is 10 to the 10th 100? Is that 100 billion?
100 billion, yeah.
100 billion, okay, yeah.
Or 10 billion, what am I saying, yeah.
Oh, 10 billion.
I can do
okay.
this, of which 100 billion are in the cerebellum. So there's a lot, and each of those neurons is multiply connected by what we call synapses to other neurons in the brain. And there are, depending on the size of the cell, there are a few, maybe 10 or 12 synapses to thousands of synapses on each neuron. And those synapses are... they may come multiple synapses from a single input. One cell might produce 100 synapses onto its friends, or there might be one or two, and the synapses connect to each other as well as to the neurons. And then there are a great number of connections which are non-synaptic but functional, what in the literature are called ephaptic connections. So you can consider the neurons as connecting to each other by metaphorically wires, so-called axons, long pathways. And as those pathways are adjacent to one another, because they're electrical, there's crosstalk between them. And that crosstalk we call ephaptic connectivity. And there's support for the idea that that also might change function.
So the idea of a neuron firing and sending a signal to another neuron, or hundreds of neurons or thousands of neurons, is a fairly simplistic picture of the electrical activity that's actually taking place.
Yeah, the field got very, very interested in the idea that the brain is principally an electric organ, which is communicating via single pulses, which you call action potentials, that occur over a millisecond or so. And certainly that's a huge part of what we do. And because all of our effector systems, all of our motor systems, move your hands, move your tongue, move your eyes, follow basically simple electrical patterns, it's reasonable to think of the brain as being an entirely electrical system, but that's not the case. And if you consider, for example, the effect of antidepressants on the brain, most people don't think that their modulation is principally of some electrical behavior. I mean, they change what the electrical patterns are, but they do something else. Right? Mood is something else, and all of that takes place in the brain as well.
And that means there's biochemistry going on in the brain. It's not just electrical signaling. There's a lot of very, very complex biochemistry at the same time, right?
Very, very subtle biochemistry, yes.
Yeah. And that's one of the areas of study. I think you mentioned psychedelics, for example.
Yes.
How they affect the brain is not necessarily sort of having an influence on. I mean, it does have an influence on the electrical activity, but it's really at a biochemical level that it changes and adjusts and changes the way things are working in the brain.
Yeah, it's tempting to imagine the brain as an engineered system. We do that. We take whatever is the sort of canonical technology of our time and generation and say, oh, that's the answer to the brain. And it's vessels of fluid stored up that become behaviors as that fluid goes out. at the time when we were fascinated by steam engines. At the time that we became fascinated by electricity, the brain became an electrical organ. These days, people are interested in computation in the brain, but it's a metaphor. And because the brain is an evolved rather than an engineered system, it is all of those things all the time. The evolution isn't particularly interested in making high fidelity wires to connect one neuron to the other. It's interested in whatever combination of factors create a behavior that's adaptive. And so if those things leak ions or leak chemicals or affect each other, if those things end up making the animal survive and reproduce, that's good enough. So evolution loves this kind of complexity. It's just as many things out there that it can exploit in order to make a more effective organism.
So since it's in a sense growing through a process over time, certain types of structures being pruned, others evolving in certain directions, and communication pathways developing that work. What works in this environment, in this context, with these biochemistries, with this electrical signaling, what works then becomes built and moves on and evolves. So the thinking about the brain as a structure, which is like building building an erector set, or it works this way mechanically, the way we tend to think about things. That's really not getting at the actual function of the brain. It's taking a storyline and putting it on top of the very sophisticated and structures in the brain and the way they actually work.
Our minds love narratives, and our minds love teleology, and our minds love the idea of creating meaning. And we could talk about the basis of that. That's what I'm really currently fascinated by. But I just want to back up for 1 second about the physical complexity of the brain. We have about 3 billion base pairs in our genome, which means we have about 1 billion codons, of which maybe 1 to 5% are actually transcribed ever. So we have on the order of 100 million bits of information in that genome, most of which doesn't directly encode the brain. And so the number of instructions that are available to build this organ isn't anything like the number of neurons in the brain. What we have is some sort of a repeated motif. That's the only way this thing can work. We have a relatively sparse set of instructions which create an organ that has, trillions of connections, but I think it's false to think about the complexity of the brain at the level of the number of neurons. I just don't think that's the depth of complexity that's encoded by the developmental process.
Right, and one of the things that you mentioned, motif, I think that's interesting because that harkens back to sort of the concept in complexity theory of fractal structures, where you have branching structures where self-similarity at different levels. There's a similarity in terms of the first level and the second level, and so this branching fractal kind of structure is, and it's useful to think of that in terms of the blood system, the circulatory system, because you start with the major arteries and then you go down, but there's a self-similarity. So the instructions that you need in the genetic code to be able to create those kinds of structures doesn't necessarily, it's not down at the level of the, designing each of the individual capillaries as part of this process of fractal branching that makes it, the coding maybe a little less complicated than you might think, but the end structure can be much more complicated than you might think.
I guess I don't necessarily think about the physical structure of the brain as having that sort of multi-scale self-similarity. But when I talk about a motif, I talk about, I mentioned this idea of a cortical column. It's a structure in the brain of a few thousand neurons, which is repeated across the surface of the cortex many, many times. but not that many times. Maybe, a few million times, right? I mean, it's the number of processing units that are in there.
A few
Um,
million, yeah.
a few million. So the one motif that we see in the brain is the cortical column. Another motif that we see in the brain is increasingly interesting to people is what they're called grid cells. And this is something that was discovered as people studied the ability of a small part of the brain to help localize rodents in space. But we find that there is another computing motif in the brain, which is heavily represented. Many parts of the brain have this grid-like thing, and those two computational motifs are capable of doing entirely different kinds of computations from one another. And we don't know how many such motifs there are. We certainly have seen that those are two dominant ones in the brain. So evolution came up with a structure which handles a certain kind of problem. And the easiest way for evolution to evolve more functionality is to just make more of them. A finger's a good thing. 5 fingers are, they're all basically the same, and it's useful to have 5 of them. The columns, the grids, you just make more of them. That's the way you get a more powerful machine.
Mm-hmm. So they're a proven technique. We might as well repeat it over and over again because it works so well. It's kind of like
right.
the biological approach. Yeah,
that's
yeah.
right. Just like the circulating hormones and chemicals, you have something called vasoactive intestinal polypeptide, which, works on the intestine and changes the blood flow there. But it turns out it's also a signaling system in the brain, the same
Mm-hmm.
protein.
Mm-hmm. Mind-boggling, to use a metaphor that may apply. So talk a little bit about how does the brain actually function in terms of computing and processing and stuff like that? What have we learned about that part of the brain? I know I had a guest, Dr. Justin Youngie, who has talked a little bit about systems within systems that communicate to each other. His specialty is in vision, so he talks about the way the retina communicates with the optic nerve, which communicate, and there's multiple levels, not just a single direction of the way the processing works.
Yeah, we certainly understand. One way of understanding the brain is to think about a layering where a given set of neurons communicates to a smaller number of neurons, or let's say that one neuron receives inputs from tens to thousands of other neurons, and that neuron has kind of an integrating function. It receives all that information, it weighs the inputs of each of those neurons, and its output now reflects the weighted inputs of those things. And you can layer that, and what will happen is that at each different level, you have a different abstraction of the data that's coming in from the first level. So if the first level is, say, let's imagine, which is roughly true, there's a one-to-one correspondence of photoreceptors in the eye to some part of the brain. So that part of the brain will receive a pretty direct representation of the light receiving the eye. Some other set of neurons can look at maybe every 10th neuron in that first layer. And what it does is produce a low resolution picture of things that repeat in the visual field every 10 times, right? And that at some level becomes a Fourier transform. So it's the spatial frequency of information. And it turns out that spectral analysis is a massively important part of how the brain receives information. And this kind of layering information is clearly a general structural organizational plan of the brain. It's also, of course, the way people have decided to do machine learning now, in part because the machine learning produces answers that mimic our perceptions.
Let me go back to a second for the neuron itself, which has these multiple inputs coming in, and it's I don't want to use a metaphor of deciding, but it can either fire or not fire. It's got an option, but that's is that a deterministic threshold? Like if it gets 10 inputs, it's going to fire, but if it only gets 9, it's not going to fire. Is that deterministic, or is there?
Well, classically, there's an understanding that if the voltage across the cell membrane exceeds some certain level, or actually is reduced to some certain level, the neuron will go ahead and create an action potential. So deterministic is a funny thing, right? It's, there's noise, there's all kinds of fluctuations. But it's not anywhere near as simple as if 10 inputs fire at once, you get a action potential out. Because first of all, if those inputs are offset in time by tiny amounts, it greatly changes the probability of you having an action potential. But then there's the question of the local electrical environment of that neuron. So whatever is the voltage across the cell membrane depends in part on the voltage outside of the neuron. And so we have large fluctuations in the voltage outside of the neuron, which are reflected in, for example, the human EEG, that modulate the probability of firing. So there's a lot going on there. And of course, there are chemical pieces of this which change the probability of firing. So is it deterministic? I don't know. There's almost certainly quantum level properties that that affect this, and therefore it becomes stochastic rather than strictly deterministic. But it certainly isn't simple.
That's an excellent answer. Thank you. And it does get us down to the fine graining of realizing that there are quantum processes that will have effects on the way the way things move up the chain within the brain. So
now, to be fair,
yeah.
Nobody has directly detected quantum effects on signaling in the brain.
Right. Roger Penrose and Stuart Hameroff have theorized about some of that stuff, but so go back to the system, the nesting of systems and that interaction. There are some ways that discrete neuronal firing gets sort of filtered as you go up, filtered or interpreted. How would you describe that process of kind of going up across those systems?
What we've seen is that if you look at different layers of this process, you tend to find more abstracted representations of what's coming in. So you can find neurons, in fact, you can find many neurons. There's a place you can find them very easily that respond quite specifically to faces. And those neurons are clustered together in a small part of the brain. And those neurons could only do that trick by having some layer before them that found maybe eyes, and some layer before them that found maybe attachments to other parts of the body and colors and all kinds of other things. So the ability of a single neuron to be responsive to a very abstract process like that is certainly a reflection of the layering of the nervous system.
Right. So each system will sort of interpret or abstract and then send it up to another system that abstracts and sends up to another system. So that sort of describes this moving up the chain. But that's not just as I understand it, not just a one-way flow. There's interaction between layers in that filtering, and what's coming in and what's being filtered, and there's a processing that goes on as well. Some messages are coming down, or something's coming down to saying, look for this or look for that.
Yes, that's right. Evolution, again, it's not engineered, so whatever works. And in our brains, there are pretty much as many outgoing as incoming connections. And that goes right down to the level of our sensory organs. There are a lot of projections from the brain to the eye. There are an enormous number of projections from the brain to the inner ear. And so the brain is centrally changing what's coming in and modulating it. So you can, at the level of the eye, you can think about how a camera works. And we have a diaphragm in the camera that changes the aperture depending on how much light is coming in. Well, that's not the photosensor in the camera. The photosensor reports to some other part of the camera that goes back and says, oh, there's a lot of light here. I'm going to change things. So we do that all over in the layers in the brain.
Yeah, so it's a intensely interconnected process. There's lots of feedback, communication going both ways, down system as well as up system, and so it's a fairly complex network of systems and processes that are kind of working together to figure out, where the body is, what the digestive system is at, where all these pieces are at, and then how does it get pulled together?
Well, how does it get pulled together? I want to go back a step and talk just a little bit about something else, and then I'll come back to this pull together question. This notion of layering is pretty well captured by the computational neural networks that are used in machine learning. And the feedback process of one layer talking back to the layer that feeds it is very well captured by the idea of a convolutional neural network, which is kind of the rage these days. And these processes, to me, are fascinating because the objective function that we choose in order to decide whether a machine learning algorithm works well is simply how well it models what we see and do in our own perception. And what we're doing is gradually building a computational system that mimics the activity of the brain, and it turns out that that helps us to better understand the processes of the brain. All right, so maybe you can reframe your question so I can unpack it a little bit and understand it better.
Well, what I'm aiming at is there's an awareness and attention, a consciousness in the system, what we all conceive of as ourself and what we're aware of and perceive and think. And, that sort of has to sit in some sense at the top of the pyramid of these systems and subsystems, because we can't possibly be aware of or conscious of all of what's going on throughout this, amazing, complex process.
Okay. So personally, I don't believe that consciousness sits at the top of anything. It's a heuristic that we like to apply because that's what it feels like. But in practice, that doesn't seem to be what's going on. So, for example, one of the most profound and repeatable properties of the brain is that if we have lesions in certain areas, we simply become agnostic to whatever that region would do. There's no conscious knowledge of it. So a simple form of it involves what is called cortical blindness, where a portion of your brain in the back of your head, that portion of your brain which receives input of your eyes, might become damaged due to blood flow problems, strokes, or whatever. And that will wipe out a portion of your visual field because this back of your brain has a one-to-one correspondence with the light detectors on your retina in your eyes. And so if I wipe out a portion over in the back of my brain on the lower right, I have no sensitivity to information coming from the upper left of my visual field. People with that kind of cortical blindness are simply not aware that that portion of the world exists. They don't worry about its absence. It just isn't there. And we see a lot of even more profound versions of this. For example, you can wipe out a portion of the brain that's involved in the sensitivity for physical motion, and people become unaware of time evolving in the normal sense. So the consciousness, if you will, of those processes is probably resident within those particular brain regions. Right? Visual consciousness is in some way resident, I think, within the visual system, and some other kind of consciousness is someplace else. But this one of the features of consciousness, the way we experience it, is that it is what's the word I want? It's focused. We could be conscious of kind of one thing at a time, and all of the other stuff is somehow not part of our continuum of consciousness. And what you've
Right, the
referred to,
continuum. So we have the feeling that our consciousness is a continuum, a continuous process through time, and it's kind of like it's only there when we're paying attention to it. We sort of think of it as, okay, that's what we're conscious of. But you're saying that there are other systems of the brain that are continuing to function even if we're not aware of it. And if we've had a brain injury where the connection to what is conscious is disrupted, that function may still be operating, but it's something that we now can't be conscious of. Is that
or it may not be operating at all, and we therefore can't be aware of it. So,
Yeah, I was thinking about something I read recently where people with brain injuries couldn't consciously do a task like writing something, but their body could still do it.
yes, that's right. There have been some amazing experiments where people who have a form of cortical blindness can still perform all kinds of visual tasks. They just can't be aware of it. So you can put stuff out there in the world that they can grasp, but they can't see.
That sounds like one of these resilient properties of the evolved human brain that you can miss the consciousness of something, but the body is still able to do it because that's a resilient piece of the puzzle.
And it begs the question of the functional significance of consciousness. I mean, if you could do the task without being conscious, there's you could become a philosophical zombie, behaving like a human but not having an inner life.
Well, that raises an interesting question. Maybe we'll get into it. I want to see if there's anything about the kind of the processing of the brain and the structures, the nested structures that other aspects of that you'd like to touch upon before we move on to that other question.
I'm impassioned by the concepts of sparsity, and I think sparsity relates in a wonderful way to the question of complexity and the focus of your podcast. There has been an unbelievable amount of progress in the mathematics of understanding sparsity, and I'd like to unpack it for a couple of minutes if I have the time from you.
Please. Yeah,
okay,
please.
So let's start off with the idea of image compression. I can take a picture with my iPhone, and my iPhone, I don't know, I mean, on the order of 10 million pixels, I don't recall right now, each of which has on the order of 3 bytes of data, one for each color, and it gets reported to some computer and saved. But the file that I save is not 30 million points ever. Typically, the file that I save is maybe 2 million points, and it's a so-called JPEG compressed image or PNG compressed image. And I look at that picture, and as far as I'm concerned, there's nothing missing, right? Even though I've clearly thrown out 90-odd% of the information. So how does that work? Well, we have computer algorithms that are mostly spectrally based. That is, mostly to look at the frequencies of information in the data, how many bars per degree of information is coming in from there, so-called spatial frequency. And an analysis of the data coming in from the camera shows that, in fact, there are a limited number of frequencies which have energy above some nominal background noise. So what it's telling us is the information coming in visually is relatively sparse. And it then became apparent that you could throw out all of the data from the camera below some threshold and still have a perfectly acceptable image. And that's the basis of JPEG compression. It's also the basis of MPEG compression, MP4, all those other things, that the spectral energy is what we kind of need in here. So then you can take this reduced set of information and reconstruct the full picture, absent whatever was in that background, which is typically interpreted as noise. So people then ultimately discovered that you don't need to acquire all that information in the first place. So if you're going to throw away 90% of it, why bother taking 30 million data points? And people have built what was now called compressive sensing devices with as little as 1 pixel that produce a full, satisfying visual image. So this is, to me, really quite profound, because it's also telling us that even through our sensory apparatus, which is incredibly limited, right? 1 octave of light, of electromagnetic spectrum becomes visible light. It's incredibly limited, our sensory apparatus. Even within that, most of the information doesn't inform us, because I can look at a compressed and an uncompressed, and a raw image, and they do not look different at all. So what this is telling us is that we sparsify the world and then reconstruct a much more dense world from this very, very sparse information. We fill in all the missing data points and imagine that they're connected in some unified whole. And that process, I think, goes on at every level of abstraction in the brain, where people know about the visual blind spot. There's a part of our eyes that simply doesn't respond to light. But people see things in that blind spot by filling in what is the most likely repeated pattern that would go across that blind spot. That's at a really low level of abstraction. But we do the same thing when we see a complex social situation which is ambiguous. We see somebody with a hood over the head, and somebody else with a gun, and somebody else who is climbing into a car and being pushed in, or apparently pushed in by
some little badge
else.
on some little shiny piece on a. Yeah.
Correct. And we build a story. And we, not only that, we can't help but build a story. It's the only way that we can manage that scene. We cannot look at it objectively in the sense that we look at the raw data and don't attach some sort of teleology to it. That process, to me, begs for this question of the cellular motifs in the brain. That is, evolution found that it was really quite useful to do something analogous to a Fourier transform in order to pick out useful information in the world, and it built a tool for that. And evolution now knows how to make that tool, and it just repeats it someplace else. But that filling in tool now becomes a whole mode of operation for our brain, and it's a whole mode of operation for our consciousness. And we imagine a much more filled in world than we could possibly detect. And all of that information which didn't come into the brain is out there, and we imagine we perceive it, but we don't.
But much of that information. Well, I want to back up a second to the issue of compressibility, because from a mathematical standpoint, there's a limit to how much compressibility there is in the information that's contained in a data set.
No, there's not.
Well, it's Kolmogorov complexity is the definition of what is the simplest algorithm that will be able to reproduce the series or the sequence. So there's a mathematical limit to compressibility, unless you're talking about and contain all of the information. If you want to keep all the information,
compression, right? But I'm not talking about lossless compression here, right? Our compression is lossy. And so, at some level of abstraction, all of the information from all of the photoreceptors in our eye could be usefully reduced to 1 bit. There's light or there's not light. And for some organisms, that's sufficient. And for much of the decision making we do, that's sufficient.
Right.
What's fascinating to me is that it's very, very clear that the overwhelming majority of the information that gets into our eyes is not even processed. It's
Yeah.
just ignored.
So this is an example of an evolved biological system. Uh, I think the word parsimonious might apply. It makes use of only what is necessary in order to benefit the evolutionary,
Right.
trajectory of this individual, this species. So
To make
survival is a
more
matter of,
babies.
survival is a matter of being as efficient as possible
That's right.
in making use of the information that is available coming into the sensing part of the organism. You only make use of what you absolutely essentially need to make use of in order to be able to react appropriately and survive in the environment. That's so that principle of sparsity or parsimonious, that principle is built into all of these systems, and especially, you're pointing out, in the brain and the nervous system.
I don't know that it's especially in the brain and nervous system. I mean, we have taste detectors for the things that inform us about the things that are useful for us to know about the world, and whether or not we could sense those tastes or smell or anything else. This sort of efficiency is absolutely a principle of evolution. And when we think about even the senses, we are aware of animals having a broad array of senses that we simply don't have. So it's very clear that biology can build a machine that can detect the polarization of light because birds can do it. It's very clear that that biology can build something that can detect electrical fields at a distance because fish can do it. And biology builds infrared detectors and incredible numbers of smell detectors that we cannot even begin to make sense of because we don't have built into our system a knowledge of those things, and a knowledge in what I tend to think of as evolutionary knowledge. That is, our system physically is aware of these things around us that are important to our
Yeah,
reproduction.
yeah. So I want to ask you about the comment you made about the narrative aspect of this, in terms of the compressibility and the sparsity and pulling information. And it sounds a little bit like sort of painting a picture out of very little information.
Mhm.
And so expand on that a little bit. And then does that go all the way up to our conscious, our conscious activity is really making a story about what it is that we're getting an input. So it appears to be continuous. We perceive ourselves as having a sort of this continuous identity. We identify that with our visual field, but it's really a narrative that's created by us to make sense of what's coming in.
I think you've expressed it just as I would. I mean, I think that's exactly what I'm trying to say. To me, this arises from at the lowest levels of information processing, evolution found a way to do this filling in, right? We can see the filling in at the level of the photoreceptors in the eye. And we can see the eye sparsifying information that it comes from the visual world. For example, the eye takes continuous light data and forms lines because of the mutual inhibition of adjacent neurons. I mean, go right into this pretty easily, but what that does is produce an edge detector, and the edges are what's presented to our brain. We abstract information from the world by sparsifying it all the time. But we also enforce on the information that we receive some level of continuity. And that expression of continuity is a motif of information processing in the brain that is almost certainly expressed in a motif of cytoarchitecture of collections of neurons that do this thing. So if you have a continuity detector, say in the visual field, that is built out of a cortical column that knows how to do that, the brain can build another cortical column that receives information from these lower levels and enforces some different abstract continuity on it, right up to whatever it is that we think of as conscious perception. Um, so, we see that all the time. For example, if we try and look at our dreams, somehow the individual events in those dreams are very, very often just sort of random seeming, right?
Mm-hmm.
But the dream itself doesn't have that experience to it. It's as though all these bits and pieces of your information become connected.
Yeah, and when we wake up in the morning, we think about it. It's just a little too weird to hold on to in some
Yeah.
senses. A different mode of consciousness that no longer works when we're awake.
Which tells us that there are a lot of ways that we could interpret that world. Um, there's the LSD experience.
Yeah.
So in my youth, right? But one of the often reported phenomena from LSD is that people perceive a connectedness to events in the world that's different than their ordinary life.
Mm-hmm.
That when that guy on the stage hit the drum, it's somehow connected to the color of the hair of the woman next to you, and it's perfectly plausible to you in the moment. Um,
Mm-hmm.
and what that kind of tells us is that there are more or less an infinite number of ways of connecting all of the events in the world to some sort of a narrative. But even under these conditions of profound disruption of normality, there still is this enforcement of continuity, you know?
So how do you make a distinction between a hallucination and a real perception? How does the
You
brain do
don't.
that?
You don't.
Uh,
I mean,
okay.
there isn't a distinction. You could go back and say something that we canonically call hallucination is more or less something that is not adaptive to us, and it's a testably false um, version of the external world. It's testably false
Mm-hmm.
because if I
Mm-hmm.
believe that there is a pond of water at the edge of
Mm-hmm.
this cliff, and I walk over it and fall down a cliff instead of put my toes in, that's a
Mm-hmm.
testably false
Right. So,
hypothesis.
so two principles. One is it's adaptive. Is it an adaptive perception or narrative? And the second is it testable. And testable narratives is what science is all about.
Mm,
Yeah.
yes, yes,
Yeah.
but certainly what we have as a conscious experience of the world is hallucinatory. I
Well, let
mean,
me ask a follow up on that exactly. I think you're probably familiar with the Libet experiments about reaction times and conscious awareness of things. And part of the speculation or the claim of those experiments were that since the actual functioning of the brain is going along faster than it takes the brain to be conscious of the things that it's experiencing or doing, that's an argument that we do not make decisions on the basis of conscious rational choice. In fact, those choices are made, and then the conscious mind makes up a story about those choices. So it's
yeah,
kind of
that's
a
interesting. It's
denial of free
interesting.
will is sort of the denying free will, because it seems to be a deterministic process that happens before we are aware of it in the conscious mind.
So there are a million profound results of the Libet experiments on time. Right? I mean, they're just multifaceted. But I don't think that I'm comfortable with saying that all of consciousness is ex post facto. In fact, we can plan things well in advance of doing them, days in advance of doing them, and then execute that plan. And that that's certainly expression of the idea that conscious awareness can do things well into the future, and it's not just ex post facto.
Mm.
Our conscious awareness of what decision we somehow made, I don't know if that has a lag or not. I don't know if that means anything anymore. So suppose I'm planning my next ski trip today,
Mm-hmm.
and in my mind I'm going through the various things that I'd like to do there. And certainly the motor process of sitting down and buying the ticket happens well after I've thought about it. But whether I was aware of having made the decision at the very same moment I made the decision.
Yeah. Yeah, that's a comfortable answer. Thank you. Thank you. I want to change gears a little bit because we're getting close to the end of time
That's too
here.
bad. I'm enjoying
And
this.
I know it's too bad. I am as well. And that relates to okay. So we have this situation where the brain is incredibly complicated. It does these does the compression sparsity. It's multiple systems. Are we going to be able to get this figured out, or is this something that we're kind of always going to be chasing something? It's never going to have an answer. I mean, I know from my mathematical guess that we're never going to have a computer model that exactly mimics the brain because there's not enough computer power in the universe. But are we going to understand enough about this process to say, yeah, we understand how all this works?
Oh, absolutely not. I mean, it's interesting because I just had this conversation with one of my colleagues in Germany last week, who is pretty much convinced that we'll solve the brain in his lifetime, and he's 10 years younger than I am. And I mean, it's just not even possible. It's in so much as, let's go back to this question of evolution and having built a brain that's only capable of certain things, right? And among the things that it is simply not capable of is understanding what it's unaware of. And we don't have the tools. We don't have the intellectual tools. Even if we end up with the instrumental tools to measure every physical process that we can find, that's not going to give us the tools to conceptually understand
Yeah.
what we have. I don't think we solve the brain ever.
Yeah. So the follow-up is, how do you feel about that? The fact that there is something that is potentially knowable, and we can't reach it, we can't know it, and you're working in this field. So is there a sense of how do you feel about that?
I'm in love with it. I'm in love with it. It's many people want to say that science is so fundamentally reductionist that it takes the wonder out of the world. But that's exactly the opposite. What you do is you look and look and look, and you get more transported by the beauty of the system and the elegance of the system. And yeah, I mean, I love the infinite complexity of the brain.
Oh, that's great. Do you have kind of a spiritual vision or a religious tradition that you feel comfortable with, or that sort of
No.
fits with that?
No, there's a philosophy. I'm not going to call it a religious tradition that's associated with Buddhism that speaks to me, but I'm not. I would never be the kind of Buddhist who writes little messages on a piece of paper and burns them in order for the spirits to whatever with them. I mean, there's a magical part of that religion also, but no, there's sufficient magic in the world as I understand it as a scientist to keep me busy and spiritual for many lifetimes.
Yeah. So now a final question. Have you experienced anything? Well, I guess if you took LSD, you might have some events in your life that seemed mysterious or transcendental, or gave you a sense of this I don't necessarily understand it, but it's really important. It's been important to my life. You have an experience you'd be willing to share?
I knew, of course, that this is one of the questions you like to ask of your guests, and I really searched for it. I mean, I haven't seen UFOs. I haven't been touched by, what I perceived as ESP. I mean, those things haven't happened to me. But I've found many, many, many experiences in this life that tell me that it's a great deal richer than my ordinary day to day. And yeah, you mentioned hallucinogens, dimethyltryptophan, DMT is one of the most profound experiences that I've ever had in my life. In the end, in helping me to better understand the brain, right? I mean, it's a physical interference with the processes of the brain. It does very specific things to perception. Those very specific things that it do to perception inform me about how the machine works. I mean, I love that stuff. And at the risk of being cliche about this, everything about the world is mysterious, transcendent, and mystical. Everything, and even the fact that we can think about it is transcendent and mystical, right? Rather than just detect it.
And we can think about it, and we can talk about it, and we can communicate with each other. Even in the face of sparsifying and parsimonious patterns in the brain, we can connect with other people.
Even this concept that there is such a thing as other people, which we understand as being different than other bodies, right? I mean, that's a remarkable feature of the world we either live in or create in. I don't know which.
Yeah. Mark, this has been a fantastic conversation. I really appreciate your time and your expertise and your willingness to share, and maybe together we'll make sense of complexity. Thank you.
Well, it's a pleasure and an honor to be part of the series of really interesting people you bring to your podcast, and I had a lot of fun.
Great. Thank you.
Thanks for joining us on this episode of Making Sense of Complexity. That was cool. Our next episode will feature Dr. Klaus Landsman, Professor of Mathematical Physics at Radboud University in the Netherlands, and winner of the Spinoza Prize in 2022. In this episode, we'll explore the complexity of quantum physics and the mathematical innovations it has inspired. So to be sure to subscribe so you don't miss that one. In the meantime, please explore the websites of our collaborators, Complexity Adventures, Planksip, and Talk of Today, and join the conversation on our social media outlets or on spiralinquiry.org. Stay well and have a great week.